Yeah, okay. All right, so this will be an introduction to resting state fMRI. We're going to talk about some basic concepts, so what is it, ways of analyzing the data, some of the main issues and challenges with interpretation, and please stop me if there's anything that you'd like to discuss during the presentation. So I thought we'd start with a comparison to task-based fMRI to build on the previous lectures. And so when we use tasks, usually our goal is to understand how particular functions are carried out in the brain. So we want to carefully design the appropriate conditions and study the activity that occurs specifically in response to that manipulation. So here's an example of an activation map showing areas with significantly higher bold signal levels during, in this case, a right-handed finger tapping task compared to no finger tapping. But one thing that you can notice is that even in this fairly well-behaved voxel, there's still a lot of signal changes going on that don't seem directly related to switching the task conditions. And we can actually do something to visualize. We can visualize the data as a movie. So we can look at all the voxels during the task and see what their activity looks like. And so here, each frame of the movie corresponds to one TR of our scan. So this is sped up quite a bit from real time. And you can see that most of the brain is still clearly active. It's just that these other regions don't go up and down according to our block design task, and so therefore they don't make it into our statistical map. So all of this other brain activity that can't be specifically attributed to the task or any other known condition is known as spontaneous or intrinsic activity. And this is actually where most of the brain's energy consumption happens. In fact, it's been shown that if you just add a task, then that only adds a tiny fraction above this large baseline energy consumption. And usually we treat any non-task related activity as noise, but there may be a lot we can actually learn by instead choosing to focus on this spontaneous activity itself. And this is basically what's motivated the field of resting state fMRI. So the way a resting state scan goes is we typically don't use any task or stimuli. Often we'll just provide minimal instructions to the subject, like to keep your eyes closed the whole time or to just keep them open but fixating on a screen. Try not to fall asleep, even though that can be rather hard. And these are just to try and control for some just pretty obvious factors. And then if we turn to the data, what we end up with is a time course from each voxel that reflects spontaneous activity, basically anything that's naturally going on in the brain while we happen to scan it. But now we have to think about how we're actually going to go and find useful information in this kind of data. And this might not seem so obvious, and just to foreshadow a few of the challenges, one is that we don't have specific conditions to compare the activity between, like we do with a task. We also don't know what the subject is actually thinking or feeling across the scan. And the term resting state is kind of a misnomer. We just know it means that they're lying in the scanner, but otherwise unconstrained. And then there's the question of how we can tell which of these little signal fluctuations are driven by neural activity and which come from the many types of noise that we know is an fMRI. And this is an important topic and going to be discussed in more detail in a future lecture. But it turns out that resting state data are very rich and interesting. But we just need to think about other ways of analyzing it. And one way is known as functional connectivity, and this is pretty much the core concept behind most resting state research. And the main idea is that we look for similarities between the time series of different brain areas. So in this cartoon, these three areas express very similar time courses, and therefore we say that they have high functional connectivity, and so do these two. And kind of some motivation behind this is that if two areas are showing similar activity, then they might be directly or indirectly interacting with one another, and this could be a way for us to learn about how regions are working together as networks. Although it actually turns out that interpreting what correlations and other measures of functional connectivity actually reflect is quite challenging, but this is at least one reason we might start looking at the data in this way. So what do you think we would find if we look at functional connectivity in resting state? Would we actually expect that there are areas of the brain that show consistently strong correlations with one another, even when we have no task? And this was the first important resting state functional connectivity study in MRI, which basically started off the field. And they found that there are correlated areas in the resting state, and importantly, not just any areas in the brain, but the ones that we'd expect to have some type of functional association. And in this paper, they focused on the motor system. So over here, we have a standard activation map from a bilateral finger tapping task. And over here, they also collected resting state data with no finger tapping going on. And what they found was that these same motor regions were preferentially correlated with one another. So in other words, if we think about taking a voxel from one hemisphere of the motor cortex and pulling out its time course, then it shows up most strongly correlated with the contralateral motor cortex and supplementary motor area compared to other voxels in the brain. And so this was a demonstration that areas that operate as functional networks also tend to correlate with one another in resting state scans. And they and others found that this generalized beyond just the motor network. So, for instance, it was found that bilateral auditory cortices are coupled in the resting state activity, so are voxels within the visual cortex, and even prefrontal and parietal areas that usually activate together in high-level cognitive tasks like working memory. And this figure was from a study where they took a large meta-analysis of task studies, and then they also took a separate database of resting state data, and they showed basically side-by-side how well-matched the task activation maps are with the resting state correlation patterns. And to briefly discuss some terminology, these patterns are referred to in the literature as resting state networks or intrinsic networks. And they're regions that make up each one of these network pictures is described as a node. And when we look at around this level of spatial granularity, meaning about the size of blobs and with standard fMRI imaging, then we tend to see about 10 to 15 or so reliable patterns that emerge from the data. And the field actually gives these names like the auditory network or the visual network, et cetera, based on which functional areas they most overlap with. However, it may be a little misleading to just think about there being a fixed number of networks running around in the brain, because we can choose to study network organization on different spatial and temporal scales or levels of detail, just as we can with refining different types of task paradigms. And so we can take any one of these networks and think about subdividing it even further into more tightly correlated subnetworks. And so that's just something to keep in mind. And illustrating how stable these patterns can be across runs, this was a study where they scanned the same subject over many, many different weeks, and you can see that in these 3 different network patterns, it's really reliable over time. And here they compared the patterns from several different subjects, each for 2 different scanning sessions, and again, you can see a lot of consistency. We can also see these types of patterns under a diverse range of conditions and conscious states. So we see them during sleep in infants, even in anesthetized animals, monkeys and rats. And one thing that this suggests is that the correlations we are seeing are not just fully driven by conscious activities like thoughts or sensations. And as we'll discuss a little bit more later, these activities do impact the strength of correlations we'll measure at any given time or a scan. But this suggests that, at least in part, there are other factors shaping these correlations, perhaps relating to the structural architecture and how that constrains the flow of neural activity, or perhaps these networks are also participate in or are driven by more homeostatic or autonomic types of functions. And this pattern I wanted to discuss for a second because it comes up a lot in resting state literature and was really important in the historical development of resting state. It's known as the default mode network, and a major feature of its areas is that they tend to show higher metabolic activity during more passive resting or fixation conditions compared to more cognitively demanding conditions, unlike most other regions of the brain. And so this observation was important for one reason is because that it caused people to rethink this idea of a resting baseline state, because there are actually certain patterns that are going on. We can't just turn the brain off. And so this is now what we study entirely with resting state.
Can I ask a question?
Yeah.
For these resting state scans, is it typically spin echo or gradient echo?
So that will really depend on the type of study you're doing. Mostly they're conventional resting gradient echo acquisitions, but if you're looking at maybe very high resolution or high field, then there may be reasons to look at spin echo. So in addition to these pretty large networks we've just looked at, you can also trace out functional connectivity patterns at finer spatial scales, and you can see agreement with known anatomic and functional organization there as well. And in this study, they looked at regions of the cerebellum and showed that these different subregions had the strongest functional connectivity in resting state to areas of the cortex where they're known to anatomically project. And here they found that you can essentially divide up or parcellate the thalamus into different functional subdivisions based on how strongly each time course matches that of the different cortical areas. And so one implication of these types of findings is that we may be able to use functional connectivity as essentially a proxy for structural connectivity. But this only works to some extent because there's not an exact one-to-one relationship between functional connectivity and structural connectivity. So what all of this points to is that even in the ongoing, really unconstrained activity across the brain, so even though this looks really, really complex, it shows a high degree of organization in terms of temporal coherence between regions. And these structured correlation patterns align really well with functional subdivisions, and that suggests that we may be able to use resting state data to map out this network structure in an individual without needing to have that subject actually go through a huge battery of different tasks. And so that's a very powerful idea. And one recent study that illustrated this point further was they looked basically at individual subjects' task activation maps. So this is from 3 different subjects, their actual activation map for a particular task. And they found that you could predict the fine details of that pattern based on information that was contained in the resting state data. So as a few implications, one, I just mentioned, we may be able to derive an array of different networks from just a set of task-free data. And if we don't need a task, then we don't need to worry about things like subject compliance. We can study these functional networks in people who may not be able to perform tasks like certain patient populations. And this may give us a very powerful tool for studying disease-related differences in functional organization. And so these are a few reasons why resting state has become a really big topic these days. And to give you a sense of how widely this is being used in clinical studies, this is a survey from 2012 showing a number of disorders where people have been using resting state to try to get more insight into different neuropsychiatric disorders. So everything from schizophrenia to amnesia or learning disabilities. So next, let's go into some methods for analyzing data. And today I'm just going to focus mainly on these 3 popular methods, but some of the later lectures this week and beyond will extend this to more interesting new approaches, and then talk about some issues with interpreting the results of these analyses. So seed-based correlation. This is a very straightforward, very popular method, and the idea is to look for areas that are most correlated with a specific region of interest, which we call the seed. And so the way this works is that you'll choose a seed, and you'll take its time course over the scan, and you'll correlate that with the time course from every other voxel in the brain, and you can visualize the strength of each voxel's correlation with the seed on a map. So the bright regions are the ones highly correlated to our chosen seed region. And then we can statistically threshold it to get to the areas that are significantly correlated. And this can be implemented with pretty much the same linear regression framework as we use for task-based analysis, but here the regressor of interest will be the time course from that seed region of interest rather than your task regressors. And as some examples, in the study, they just picked some different seeds, and you can see what kind of correlation maps result. So if you take a PCC seed, then you'll get a pattern that looks like the default mode network. Here, an intentional network, motor network, and visual areas. So these were kind of the canonical resting state networks we've just discussed. And this is if you do it at a bit more on a more high resolution or finer scale mapping. And here they placed seeds in different regions of visual area V2 that correspond to different eccentricity representations. And what they found was that these, for each, the red areas indicate the other areas of the brain that are most highly correlated to each seed, and it tended to find similar areas that have a similar eccentricity representation as the seed. And so this here is a comparison with their actual eccentricity map. And so this is all done with the eyes closed and resting. So even in spontaneous activity, this again shows how well the functional connectivity can reflect topographic organization. And AFNI software developed here has a nice exploratory tool for seed-based correlation analysis called InstaCore. And the way it works is if you click on a voxel, then it will magically show you instantly the seed-based correlation map from that voxel. And this can be really helpful for initially exploring the correlations structure in your data, like from different seed regions. You have to avoid fishing for any kind of effective interest. And this can also be a nice way of kind of quality checking your data, because sometimes if you click on certain areas, you can see large artifacts popping out really obviously, so that can be useful for that too. And some example questions where you might use seed-based correlation. These are along the lines of, if you're looking for, are there any areas across the brain whose functional connectivity with your seed is significantly different between two conditions? Or are there any areas whose correlation with that area may increase as the severity of the disorder increases? And this was an illustration of that, showing two areas that became, in their data, more strongly correlated with a measure of psychopathology. One of the considerations we need in our seed-based analysis is where we want to place that seed. That's kind of the main question. So once you've kind of narrowed down the general area, then you have to ask, would you choose an atlas-defined region, or maybe you want to go with a coordinate from the literature, or maybe you want to actually carefully draw it on the subject's anatomic image, or have them do a task localizer, essentially, to try and find that coordinate. And these questions should be really guided by the your particular study and hypothesis. And the results that you can get from a seed-based analysis can be really sensitive to exactly where you place the seed. And so if you move that seed too much, like beyond the intended area, you can end up in an actually different functional area that seeds a different functional connectivity pattern. And so this is an example of that, moving a seed in the parietal cortex from one zone to another, and showing, you can see how the correlation maps change. And this can give you unintended variability, because especially like if you're between subjects registration is off, then where that ROI lands in your particular subject might not be where you intended it to be. And so this could be an argument for trying to use more subject-specific ROIs. And in this paper, they also did something interesting, noticing that you could leverage this sensitivity in order to try to trace out functional boundaries across the brain in a subject. So if you are to find where there's a dramatic shift in functional connectivity as you move the seed, then that likely means that you're jumping into a different functional area. And here's a movie that also illustrates this kind of concept. Here they were moving a seed region around parietal cortex, and showing where for each location of that seed what the correlation pattern across the whole brain looks like. Mesmerizing. So now let's move on to a second approach, which is called ICA, and this is probably the next most popular method of connectivity analysis. And the abstract level, this black box level description is that basically you input your data, which is the sequence of fMRI bold signal volumes across the scan, and what you get on the other side is a set of maps or components, each of which can be thought of as a network, and people actually use the term component and network pretty interchangeably when they apply ICA. And these maps also come with a time series that represents the activity of that component over time. And so in contrast with seed-based method, this is multivariate. So it essentially solves for a set of networks at once. It's data-driven. There's no need to choose the seed beforehand, but you do have to specify some other parameters of the model, like how many components you'd like it to return. And the components, when you look at these, a lot of them will look like functional networks that we expect, and other ones will reveal artifacts in the data, and so this can make ICA a useful tool for analysis and noise reduction. And to look a bit closely at the model, the model is that if you, at any given time frame, what we are measuring in our original data is represented as a mixture of activity coming from these different brain networks or components, and the relative contribution of these components is something that is different for each one and can change over time as well. And so when we start out running ICA, we have only this fMRI data. We don't know the components or the weights, and so it tries to solve for this by placing some assumptions on the data, like that these components are going to be spatially independent. And the real-world analogy that's often used for ICA is to imagine that you're listening to several people talking at the same time, and you want to unmix what you're hearing into who the different people are and what they're saying. And so this is like our data, our networks that are all talking at the same time. But even though we're just using this blind mathematical criterion for finding the components, it gives us really realistic looking functional networks, and it does this with quite high reliability. And so this comes from running ICA on a group of healthy subjects, and you see familiar networks, visual network and default mode network, for example. And these are usually consistent with results of seed-based correlation on some level, which makes sense because even though there's not really a straightforward mathematical relationship between these methods, both of them tend to converge on areas that share really similar time courses with one another, which these patterns do pretty robustly. And so as a few considerations, one thing is you do have to, well, if you're going to try to say that this component is the working memory network, then you need some criterion for being able to say that. You can't maybe just eyeball it and say this one looks like working memory network. But so you want to try to come up with objective criterion, like, for example, some studies have tried to match that pattern with an a priori template, like from a task activation map or a functional atlas. Another consideration is that we have to choose the number of components that come out of the ICA, and if you end up choosing a large number of components, then you can get these familiar networks splitting up amongst many, many different components and or becoming more sparse, as seen in this example with choosing more and more components. And I'm not really going to go into this, but there's also some questions around how to best do a group comparison when you're using ICA and what is the statistics to use. And so I mentioned before that one use of ICA is for noise reduction. And ICA kind of, one nice feature is that it's got this kind of built-in denoising mechanism in the sense that it will tend to conveniently peel off certain large artifacts into separate components. And so by definition, once it's done this unmixing, the remaining components don't have as much of that inside of it. And so if we are interested in just one or two particular components, you can choose to carry that forward into your analysis. Or you might be interested in, for example, identifying the components that are noisy and removing them from your data, and then reconstructing a cleaner data set from the remainder. But this also requires some way of identifying those noise components, which I'll talk about in a second. But I also wanted to use this slide to show that it gives you a nice idea of how ICA gives you a useful visualization of the structure of your data, and it can be helpful for just looking at what patterns are there and maybe helping you discover types of noise that you might not have expected or thought to even model. So going back to identifying the noise components, there's been a few avenues pursued, and one has to do with using machine learning classification to try to learn which of these components look like noise based on a training data set. So then you might be able to later on apply it to your own data. And this program, FIX from FSL, is one program that's based on this idea. And a new technique which has been developed here in Peter Bandettini's group is known as multi-echo ICA. And Jen Evans described this a little bit last week, but briefly the idea is that if you collect data with different echo times, say 3 echo times, you can use the fact that bold and non-bold portions of the fMRI signal have different properties as a function of increasing echo time. You can use that to try to separate them. And so if you combine this with ICA, then you can form a ranking of components according to which ones on a spectrum, basically from most likely bold or to least likely bold. And this can separate many components very cleanly. Some of them are kind of on the borderline and more ambiguous. And this brings up the point that we don't, the noise and the signal can't really be perfectly split by ICA, so we don't live in this ideal world when they're really completely separate. And some of the strongest forms of physiological noise arise from a bold mechanism, and they also have large spatial overlap with resting state networks, so ICA can't always separate all of the sources of noise. And basically this summarizes considerations we just talked about. From an interpretation standpoint, ICA output is a little trickier than seed-based correlation maps because the algorithm itself is more complicated. And like, for instance, the value you get in one of those spatial maps it represents its membership in that component, not just the straightforward correlation. And I can just point you to this article for more discussion of trade-offs between these approaches and other types of discussion about methods. So the final class of techniques we'll go into is complex network or graph theory analysis. And here the main idea is to model the data as a set of nodes that are connected by edges, where nodes are often taken to be distinct brain areas, and the strength of a node of an edge between the two nodes will represent some measure of association between them, like how correlated their time courses are. And once you've specified what your nodes and edges are going to be, then you can try to characterize properties either of the whole network or of individual nodes in that network. And so at the kind of more global level, one measure you can consider is modularity, and this will identify groups of nodes that have high interconnection within themselves and are less connected with nodes outside that module, similar to communities in a social network. And focusing on individual nodes, one thing you can look at is how much that node will act like a hub or basically bridge between two different communities, like this guy here. And these are some articles where you can read more about different network measures. And doing this type of analysis also involves a lot of choices, also none of which are really that there's really an answer to, but it really depends on what we want to study. And the choice of nodes and edges here is critical because that's going to affect what we get out from our any subsequent analysis on the network. And so for taking nodes, for example, are we interested in looking at resolution of countries or continents? Or for the brain, do we want to use a big anatomic atlas, or do we want to use a more functionally based parcellation? And then if we randomly carve out nodes without really considering the underlying information or structure, then we might get unintended results because we'll be really mixing inhomogeneous entities into one node. And there's been a kind of growing subfield of fMRI methods that focuses on ways of parcellating this data into nodes. And here's an illustration applying network analysis to resting state data. Actually, this isn't really pure resting state data because they also presented a faint tone occasionally across that scan. And they were looking to see whether there was anything in the resting state brain activity before each tone that could predict whether the subject heard it. And they report that when the preceding resting state activity was organized into a more modular configuration, then subjects perceive that tone more easily than if the brain were in a less modular state. So finally we'll move to, or not finally, but now we'll move to interpretation and a lot of issues. I mean, one big thing is how can we tell, how well can we use resting state data and measures of time series correlations and other types of models to really say something about how brain regions are acting as networks or communicating. And this statement kind of captures the essence of what we can do, I think, at the moment, which is that functional connectivity is a powerful but ambiguous mapping tool. So it can be really useful for detecting interesting phenomenon, but then once we have that result, it's not perfectly clear how to interpret it. And one reason for that difficulty is that there are many non-neural and neural sources or processes that are going on that shape the individual signals you measure in different voxels and the correlations between them. And a lot of these processes are not easily measurable, or are there, and their influence on the fMRI signal is not clear. We're dealing with pretty noisy and complex data. This can be sensitive to the decisions we make in modeling and pre-processing. The relationship with structural connectivity and electrophysiology is still not very well understood, so that ultimately limits how far we can push our interpretation on those ends, and is also a research area, ongoing research area. So this is an example of a non-neural effect that can influence functional connectivity from physiological processes like breathing and heart rate have been referred to as physiological noise. And one effect of noise is that if you have a process like breathing that tends to influence many different regions of the brain in common, then this could create spurious functional connectivity. So what's shown here in these areas, these are areas of the brain found to be most strongly influenced by natural changes in the depth and rate of someone's breathing across the scan. And unfortunately, you can see that it affects many regions of the gray matter, overlapping with important resting state networks. So if you can imagine placing a seed in an area that contains a lot of breathing-related noise, then you would see inflated correlations with all the other areas that show strong breathing effects if you don't try to remove this. And there's also the danger of there being systematic differences in breathing across subjects or across populations, and so something like this might show up as increased global connectivity in one population versus another. And so we always want to try to reduce any known sources of noise as best as we can. And for something like physiological noise, the problem is definitely not completely solved, but you can make you can help a lot by recording breathing and cardiac activity during the scan and trying to model those effects out. So that's, it would be a good idea to collect that data, at least if you're able to. Head motion can also change connectivity measures too. So this is a study by Jonathan Power pointing out that if you have children showing more head motion than adults, then you can get what looks like age-related changes in functional connectivity. In general, telling noise and signal apart, this is a problem for all kinds of fMRI studies, but in resting state there are a few things that make some aspects trickier. And one is because we don't have timing information about for events of interest in order to try to help us distinguish the signal from the noise, and we can't use that to do anything like trial averaging on the time courses. And with functional connectivity, instead of comparing our data to a noiseless task activation regressor, we're actually comparing time series of noisy brain areas to one another. And so we can really get the wrong answer about connectivity between brain areas. So it's another reason that noise in resting state is kind of dangerous.
Do you know if physiological noise tends to be more higher or lower frequency?
Oh, yeah. So, there's a few different types of physiological noise, and some are pretty high frequency. For instance, cardiac pulsation is about at the frequency of the heart rate. And so when we're using our normal, 1 to 2 second TRs, then we're really undersampling that heart rate. But other things like the depth of breathing that I just showed, that's a really low frequency artifact. So in that sense, it doesn't really help you that much to try to acquire the data with the faster TR, because it's actually comes from a bold signal like mechanism, and it's very slow, and so it's less clear how to take that out using frequency information. Another issue with resting state is that it can be really hard to validate your pre-processing methods. And how do you tell whether they're doing the right thing in the first place? And so this was a pretty famous debate over whether or not certain networks in the brain were anti-correlated or negatively correlated in their resting state activity. And this was pretty interesting because if the default mode and attentional networks were really anti-correlated, it could be something very or related to their functions as maybe intro or exteroceptive networks. And so original studies that pointed out this anti-correlation, they had done a pre-processing step of taking the whole brain average signal as a proxy for this is our noise we don't care about, and regressing that out of the data, which can be shown to enforce mathematically a certain number of negative correlations in the data. And so if you try to replicate this without doing global signal regression, it's less clear whether those negative correlations are just a side effect of global signal regression, or if they actually do exist but are covered up by noise unless you really take aggressive procedures to try to remove the noise. And so in terms of these anti-correlations, I think it's been found by other ways that they do exist, but you don't get as spatially extensive negative correlations as you do if you do a global signal regression, because a lot of those global signal regression induced negative correlations are spurious. So now let's discuss a few sources of neural variability in functional connectivity. And drowsiness is one unavoidable thing, since these scans are, they're not doing anything. And so unless you're studying arousal effects in itself, then this can act as a confound in your data. And so to ask the question of what happens in resting state signals when you become sleepy, this was a really long resting state scan collected here in Jeff Dunn's group, and they were scanning a very drowsy subject and noticed that as time went on, the resting state signal fluctuations in many different areas increased in amplitude. And so amplitude increase seems to be one thing related to drowsiness, which was shown by other studies. And also here they found that in addition, there seemed to be a widespread increase in positive correlations over the brain. Here they studied subjects before and after they were given caffeine. And a similar thing found with using an EEG-based index of vigilance state, so more higher correlations between regions when EEG suggested drowsiness versus when EEG suggested alertness. And further changes occur that if you actually do fall asleep. So in the figure, they found that the anterior and posterior parts of default mode network basically became less coupled with one another when subjects were in deep sleep. And over here, they found that you may be able to identify different sleep stages based on looking at whole brain patterns of functional connectivity. And so that's kind of exciting because you may be able to do sleep stage identification just based on fMRI alone. And when these authors applied their method to a bunch of resting state scans, when people were supposedly staying awake, they also detected what looked like a lot of sleep in those scans. And so this can really change our correlation values, and it can be really important, again, if we expect any systematic differences between groups or subjects, because in resting state analysis, the effect sizes usually tend to be pretty small, and so these things can have an impact. But if we look at this from a more positive viewpoint, the fact that we can see these neural state changes reflected in our resting state signals also means that we can use that data as a way of maybe studying what kind of correlates are going on in the spontaneous activity of these natural changes, which is something we couldn't analyze the task data quite like that. So finally, another thing that influences resting state is mind wandering. all the different things that you may be thinking across the scan. And here they tried to figure out what people were thinking after the fact by administering a questionnaire, and found that basically, I think the result was imagining scenes versus doing imagined language can cause a significantly different impact on functional connectivity between those networks. And recent work from Javier Gonzalez-Castillo shows that if you now kind of hone in on smaller time windows of data, so usually these resting state scans are like 8 to 10 minutes, but if we start thinking what happens over like the 20, 30-second windows of data, here they found that they were able to distinguish based on connectivity whether subjects were engaging in one of several different kinds of tasks. So here they did have continuous tasks going on in order to train this model and figure out whether or not you could really see systematic differences in functional or in resting state or in functional connectivity between regions. And now I just wanted to spend a minute on the link with structural connectivity. So this could play, we think this might play quite a big role in shaping functional connectivity. And one way that we can study this is by looking at patients who have a disruption of structural connectivity. And here in patients born without a corpus callosum, which forms major connections between the hemispheres, they found that if they did a task, then both sides of the brain were still activated to that task, but the correlations from one side to another were greatly reduced. And here they were able to look at functional connectivity before and after they had surgery to remove the corpus callosum, and you can see a very big effect of basically bringing the connectivity from bilateral to only one hemisphere for most of these different seed regions, which is what each of these rows corresponds to. But there were some areas they found where bilateral connectivity still survived, and it was pointed out that these areas had they had connections between the hemispheres that didn't cross in the corpus callosum. So it points to a role of there being indirect connections that also mediate functional connectivity. In terms of the link with electrophysiological signals, this is not in humans. This is actually a patch of visual cortex in the cat, I believe. And they used a very direct measure of electrical activity with voltage-sensitive dye. And they were studying the spontaneous activity patterns. And they found that if they focused on certain snapshots or time frames of that spontaneous activity, then you could also see structure that looked a lot like the evoked activity. And so this whole phenomenon of there being spontaneous and a very functional organization to the spontaneous activity has also been seen outside of fMRI. But in terms of trying to link the precise resting state fluctuations measured in humans with an electrical feature of brain activity that we can measure, like with EEG or maybe more invasively with ECOG, this still remains a complicated picture. And a few things that are known is that in terms of the link between fMRI and electrophysiological signals, it's been found in several studies that power in the gamma frequency range, usually taken above 30 hertz, local field potential has a fairly good correlation with the fMRI signal. And here, using ECOG data from patients who are about to have surgery, they found that if you look at basically a seed-based correlation analysis, but instead taking gamma power electrophysiology instead of the fMRI signal, they found that you get a somewhat similar pattern of correlations as seen in that subject's fMRI data. So gamma power may indeed also may follow the same kind of functional organization, but there are also some examples that indicate correlates in other frequency bands. So one frequency band is, slow cortical potentials, which is a very, very slow frequency band, have also been shown to have a sort of a network organization as well. And there have been other studies showing a more distributed picture in terms of there being not one but multiple frequency bands correlating with the signals of different resting state networks. And some have pointed to there being a broadband effect, so all frequency bands rather than any frequency-specific relationship. And so here's a review article you can look at, and this is still also an interesting area of work. So now we'll finish up, but before summarizing, there were just two topics I wanted to just say a few more things on, and one of them is clinical applications, and as mentioned earlier, this is a really huge area since resting state may really help us understand in new ways how there are maybe network level differences between different disorders. But beyond those kinds of group comparisons, it's also being explored for areas like surgical planning, early diagnosis, guiding treatment decisions, or predicting treatment response. And here's an example of a current direction that tries to leverage functional connectivity to define target locations for non-invasive brain stimulation, like with TMS. And so, for example, one thing we can do is try to tailor our stimulation site to particular functional networks based on mapping their resting state activity. And one thing they did here was to also use functional connectivity as maybe a way of accessing deeper brain structures by finding out which of the more accessible cortical areas these most likely connected to. Another direction has been in kind of using a lot of sophisticated machine learning algorithms, like deep learning, as basically data-driven ways for finding what information, what features in resting state data may be linked to different disorders. And one thing to keep in mind when you're looking at functional connectivity as a measure of differences is that differences in this functional connectivity measure may not relate directly to some change in network interaction or some feature of the interareal communication, even though that's the language we usually use and how we are inclined to interpret it. But really anything that changes the underlying signal in a region, maybe the anatomy or neurovascular coupling or something, can also propagate into seeing a difference in the functional connectivity. And so if you find a functional connectivity difference, that may be something that may point you to looking also at other aspects of local change that might be important and might give rise to that effect. But I mean, in terms of whether or not we really care about resolving all those factors is another question, because some may just care about whether or not there's the information in resting state that can be a diagnostic tool, and maybe it doesn't matter so much exactly what mechanisms cause that. And just a final trend to highlight, and this will also be covered in another lecture later on, is that there are lots of data sharing efforts now across a lot of different imaging sites where there's databases of resting state data or resting state scans from healthy participants, also certain patient groups. These are just a couple examples. And so now this data can be mined by anyone to look for lots of new things. So that's one nice thing about resting state data is you can just try to discover new things from the existing data. In fact, you probably can even write your whole thesis now on fMRI and not have to collect any of your own data. So as a summary, resting state fMRI is now a very big field and proving valuable for a lot of applications. There are lots of methods. We barely scratched the surface of what's out there and the ways people are looking at this. It's important to be aware of their trade-offs and then try to stay as close to the actual data as you can to really understand things and look at time series a lot. I think that's really useful. And then there's lots of different factors that can affect our measurements, and we can't always control for things, but at least need to be aware of what kinds of factors could contribute and try to correct for things as best as we can. So, oops, this is all. Thanks. Any questions? Yeah.
So one of the issues of task-based fMRI is that you have this really bad signal-to-noise ratio, right? So you could like technically use your resting state analysis data to like from regressors to actually clean that up a lot, right? To like reduce noise a lot.
Oh, yeah. No, that's interesting because yeah.
Or is it too naive? I don't know.
No, actually, I mean, similar things have been done, definitely. Like, I mean, one study around like 2006 or so was showing that these spontaneous fluctuations account for a lot of trial-to-trial variability in an event-related task, and that if you were focusing on a task that affected one hemisphere and you tried to estimate the spontaneous activity looking at the other hemisphere and remove that, you can get a lot more consistency. And I think there are other ways, I mean, depending on what the goal is, then that, yeah, could definitely be interesting. Yeah.
Have people looked at functional connectivity over time? Like,
yeah.
connectivity in a seed, like connecting to another seed maybe a second later or 10 seconds?
Yeah. That's like one of my favorite topics. So actually, there's going to be a lecture on this Javier is going to give in like, I think within a week or something. It's really interesting because, I mean, that opens up the whole question of what do we mean by a network exactly? Because, I mean, we have to define that over some time period, and those resting state networks tend to be the things that couple with each other the most stably over time. But at any given instance, you can look and there's different regions that are interconnected, and just to carry out whatever function is just happening at that second. And that if you start looking at that information and the way in which connectivity patterns change, there can be a lot of information there. Oh yeah, the question was: is there consensus on what the best kind of instructions or thing to have the subject do to measure resting state? I mean, this is hard because it's not like there is a resting state or isn't a resting state. It just is, what it is, I guess. And so, I mean, I know there have been studies looking at eyes open and closed. I don't think there's really some have pointed to less variability, I think, with eyes open, but maybe because they're less likely to get drowsy. But then if you're studying drowsiness, then maybe that's something that's interesting to you. And so, and actually, there's also been a lot of work looking at spontaneous activity, not with no task, but guided by some kind of task that's going on, or looking at the interaction between the task and the activity too. So I think that's hard to answer. Right. Thank you.